Okay, so is my screen visible to everyone?
Yes, sir.
Okay, thank you. So as I was saying, today we are going to estimate the DNA concentration using diphenylamine reagent. So basically, the concept and the principle of this method is very much alike the concept that we had studied in the last experiment. That is, we are going to obtain a colored product at the end of the reaction of whose absorbance we measure at a particular lambda max, and thereby we calculate the concentration of a target molecule, which here is DNA. So what we do is talking about the principles. So here we have DNA as our target molecule, which we are going to quantify. We treat our DNA with diphenylamine reagent under acidic condition. So as a result of which we are going to obtain a blue color compound of whose absorbance we are going to measure at 595 nanometer. Okay, so what happens in this reaction, this process is, this reaction is actually given by 2' deoxypentosis of the DNA, and therefore it is not specific for the DNA only. It can also be given by these pentosis residues alone also. So what happens in this reaction is that when we treat the DNA in the acidic solution, the DNA undergoes depurination and dehydration. Depurination, that is the nitrogenous bases, the purines are removed, and the DNA chains are hydrolyzed, and it under the pentosis sugar undergoes a dehydration to form so that this 2-deoxyribose sugar are converted into a very highly reactive compound that is called omega hydroxy levulinyl aldehyde. Okay, when it reacts with diphenylamine, this omega hydroxy levulinyl aldehyde, it reacts with diphenylamine in the second reaction to give a blue color complex. And this is the complex that we use to determine the concentration of our DNA. So in this reaction, there is one thing that we have to note, that only those deoxyribose sugars which are attached with the purine, purine nitrogenous bases give absorbance. Okay, so not the other sugars. So this is we have to know. And based on this last line, I'm going to ask you a question at the end of this experiment. So please be just uh, note this line, that only those deoxyribose sugars give reaction which are attached with purines, right? So our schematic representation of the principle is here. So yeah, what happens is our deoxyribose sugar after depurination undergoes dehydration in acidic condition to form omega hydroxy levulinate aldehyde. And this is the product which is highly reactive. It undergoes further dehydration to form this product, which then reacts with diphenylamine. Okay, 6 molecules of diphenylamine react with single molecule of hydroxylevulinate aldehyde to produce a blue color compound. Okay, so this is the compound. This is the compound that we are going to whose absorbance we are going to measure, right? Talking about the reagent preparation, we have to prepare 3 reagents. First is buffered saline that requires zero point 0.1 molar NaCl in 0.015 millimolar sodium citrate to produce this buffered saline. And we have to adjust its pH to 7. Okay. Now, the second reagent is diphenylamine reagent. So what we do is we take 10 gram of pure diphenylamine. We take 10 gram of pure diphenylamine in 1 liter glacial acetic acid. Okay. In addition to concentrated H2SO4, right? Now we prepare our DNA solutions as we had done in the last experiment. What we did is we first prepared a stock solution. So here our stock solution would be 20 milligram of calf thymus DNA in buffer saline that we had just prepared in the first step. So if we add 20 mg of calf thymus DNA in 50 ml of buffer saline, what could be the concentration? Can anyone tell me?
Sir, is it 20 mg of calf thymus DNA, or it is a dilute solution of calf thymus DNA?
Okay, sorry, I couldn't hear the question. Just I need 2 minutes break. Just 2 minutes.
Sir, your voice is also breaking in between, and I am not able to. I don't know
Yeah.
if others are able to
Okay.
hear.
Okay. Hello.
Yes, sir.
Hello. My voice audible?
Yes, sir.
Okay. Okay. Sorry for the interruption. We had something noisy going around in the lab. So I had to change my location. So yeah, I was asking.
Sir, I cannot hear you.
Yeah, I'm not speaking at the moment. Yeah. So is my screen visible in full screen?
Yes, sir.
So what I was asking is, if we add 20 mg of calf thymus DNA in 50 ml of buffered saline, what is the concentration expected?
So can you say in which unit we have to tell?
Mg per
If
ml.
you say in mg per ml, so
Yeah.
0.4 mg per ml.
Okay, so 0.4 mg per ml would be our stock concentration, and we have to use
Sir,
this stock. Yeah.
20 mg of DNA will be like a very high amount of DNA, isn't it so?
Hello. You are not audible.
Yes, sir. Sir,
Can
am I
you?
audible?
Yeah. Please ask your question again.
So my question was 20 mg of DNA would be very high amount of DNA. I guess like 100 trillion base pairs of nucleotides accounts for 1 nanogram. So in that way, 20 mg of DNA will be very high amount of DNA. Like,
In
isn't
terms
it
of
so?
nucleotides and the molecular weight only. But if we
Yes.
weigh, it is actually very small. And if we dissolve it in 50 ml, it is further very small. So it's okay. We can manage. Yeah.
Yes, sir. Okay, sir. Okay.
So the next step, after preparing all three solutions, we have to start our boiling bath, because before doing all this experiment thing, first takes time to reach the temperature, boiling temperature. Okay. So after preparing the solution, the next step, that is, we have to prepare the reagent. Is my slide or demonstration of experiment? Can you see?
Yes, sir, it is visible.
Okay, so now you have to see how we have prepared the individual concentration from the stock and performed the whole of the experiment. I will be explaining the individual steps. So I'm starting the video. So first we add required amount of stock solutions into the individual test tubes. Prepare our standards, right? And now we make up the volume to 2 ml by adding buffered solution, buffered saline in all the test tubes. And finally, we add 4 ml of diphenylamine reagent in all the test tubes. We mix the contents either by vortexing or by simple shaking. And then we put them for water bath at boiling temperatures for 10 minutes. And we set the spectrometer wavelength to 595 nanometer as we did in the last experiment. And then we start taking the absorbance readings. First, we do it for blank. We set the blank's absorbance at 0. And after that, we take the individual standard concentrations and note their absorbances. Okay, so this is how we have performed the experiment. Here, wait. So what we have done is we have prepared individual standard DNA concentrations of 0.1, 0.2, 0.3, and 0.4 mg per ml from our stock, which is a 0.4 mg per ml, right? Based on these concentrations, you have to calculate what amount of DNA stock we have added into the individual test tubes and what amount of buffer saline we are going to need to make up the volume to final 2 ml. Okay. And as we have also observed in the last experiment, the blank contains everything except for the uh, test sample, right? So the blank will be containing 2 ml of buffer saline alone, along with the 4 ml of diphenylamine reagent, right? After we have prepared the individual solutions, we incubate them for boiling at for 10 minutes and note their absorbance. So according to the blanks absorbance at 0, we get absorbance of all the other samples. Plot a standard curve as in the previous experiment. So we are expecting a linear plot with the equation y is in the form of y equals mx. You also have to uh, provide their R square values in the graph with, okay, so this will be the graph of absorbance at 595 at a y-axis and DNA concentrations of standards on the x-axis. Using this graph, you have to determine what was the DNA concentrations of the unknown sample that we have used. Okay, so this is what you have to do in this experiment. It's a very small experiment. Okay, and the graph that I'm showing here is not the graph you are expecting. So please take care of this also. This is done. This experiment is over here now. But I was talking about in the first slide that in this procedure, what happens? Only deoxyribose sugars give you the absorbance, right? And if only deoxyribose sugar that are attached to the purines give you the absorbance. So the actual absorbance values and the actual concentration should be half of the DNA. So can anyone tell me why we are not doubling the DNA concentration that we are obtaining here? Is my question clear to everyone?
Yes, sir. Question is clear.
So let me take you back to this. Little complicated question for a very simple answer. So what we have here is only those deoxyribose are going to give absorbers which are attached with purines. And as we know, half of the deoxyribose are with purines and other half are with pyrimidines in the DNA. So what we are going to get from this experiment is half of the DNA concentrations, right? So why
Yes,
are
sir.
we why are we not doubling the DNA concentration that we get from this experiment? Why are we only taking the concentration that we get from the graph?
Sir, because we already know the DNA concentration into the solution while we are creating that graph, so that kind of represent the total concentration, so we can directly use the calculated value.
Yeah, exactly. So that is the right answer. So what we are doing here is the absorbance that we are getting in this case, in this experiment, is from the non-DNA concentrations. Okay. So whatever absorbance we have, they directly relate to the exact DNA concentration that we are using. And the same thing will also apply for the unknown also. So whatever absorbance is here is related to the total DNA concentrations and not to the half of the DNA concentration. So it was, again, as I said, simple answer for complicated question. So if you have any question, you can ask me. So I have uploaded the presentation for this experiment. You can go through it, and within the next half an hour, you can ask me questions related to it. Okay, and there is one more thing. Sure. So after checking your first experiments, I have found the common consensual mistake that some of you are making. So the first thing is many of students have not provided their Excel graphs. They have only plotted the uh, graph with their handwriting, right? So you need to provide your Excel graph also. Okay? That is more important than the physical graphs. Okay, so Excel graphs are mandatory. Okay, the next mistake was some of you did not write your procedures in passive voice and past tense as it was made very clear from the first experiment itself. So please write your experiments procedure in passive voice and past tense. Okay. Uh, Next was you have to write units of your parameters on the x-axis. Like you can see here, DNA concentration along with its unit, mg per ml, right? So you have to write the units also. Okay. And as you know, absorbance, no units, so it's not needed. So wherever you require to add units, and not just in the graph, but also in your procedure, also in your methods, everywhere, and also in your conclusion. So everywhere you require units, you have to write units. The next is, as I told in the last, for the last experiment, it applies to all your file submissions. Please name your files with this format. Okay, 2019 DB1, your specific roll number, 4-digit roll number, underscore your name in capitals, underscore exp and experiment number. Okay, so please follow this. Uh, Many of you were following this, but some of you still didn't. I don't know why. Maybe because you submit your experiments at the last minute. So please note all these 4 mistakes. And there's one more thing. The deadline for this experiment and all the experiments after this would be the next day, midnight. Okay. So please submit all your experiments before the deadline. Okay. So 1 day is enough, I think, for each of these experiments. Right?
Sir, actually, in this semester there are many labs, so it becomes really difficult to submit the next day. So,
So,
uh,
what
like
happened
one
is,
day in 1 day deadlines is really less.
okay, so please try to counter my next argument. Uh, the last experiment was very small. I asked for a 1 week deadline, and 90% of the submissions were given within the 15 minutes just before the deadline. So this might be because we have completed the experiment within the next 30 minutes only. So if experiment can be
You can
complete 30
it,
minutes,
so you can extend the like okay if you don't want to give 1 week for submission, but at least like
yeah, I
not
can
just a
give
day.
2 days. Two
That
days
would
might be enough.
be
2 days will be shortly enough.
Sure. I
so,
mean, we have a lot of labs and we have different submissions on different dates.
Yeah.
So 1 week
yeah, sir.
submission works for pretty much everyone. Like we have a submission for a very big assignment today itself, and then we have
So,
labs almost every day except on Thursday or Friday for some people.
okay, so I
So,
guess 2 days
So you
enough.
can
Is enough.
keep
so
it
you
on
can have
the
it on maybe on Friday, end of day or Saturday end of day maybe.
yeah, that would be better.
Yes, sir. Friday end of day would be
So
great.
I also need to check your experiments. Okay, I haven't checked your last experiment because I have received almost all of them yesterday only. So if I don't check your experiments, I don't find your mistakes and I don't tell your mistakes. And if I find the same mistakes while checking, uh, you will end up losing some marks, right? So it's only for you. I'm asking to submit me assignments earlier so I can go through them. So okay, ठीक है. 4 days, Saturday, every week, Saturday.
Yes, sir. Saturday is fine.
Okay. So Saturday midnight every week, you can submit your experiments. After that, uh, you can submit, but I will not take it into account, right? So this is all. Uh, After 20 minutes, I will take your attendance, and before that, you can go through this presentation. And if you have any questions, I'm here. You can ask.
हेलो।
Yeah,
हेलो सर, मैंने दो ग्राफ अटैच करे थे, एक हैंडमेड था एंड एक एक्सेल से बनाया था। तो
yeah.
जो यूनिट्स हैं वो मैंने हैंडमेड वाले में एक्स एक्सिस पे लिखी हुई हैं, मतलब दोनों एक्सपेरिमेंट्स में। तो इज इट सफिशिएंट और शुड आई आल्सो मेंशन इट इन एक्सेल ग्राफ?
You can also mention the units in Excel graph also. It is important, but it's okay if you have missed it. But please mention your units everywhere, not just in a graph only. As I said, units are to be mentioned. Otherwise, there is no meaning of those numbers written on the paper if you don't write your units. Okay?
Okay.
Your units are very important to be mentioned everywhere.
Sir, for this experiment, we have to give Excel graph or handmade graph?
Excel graphs are mandatory, and handmade graphs I ask because to make sure that you only have made these Excel graph and not just copy. So basically, both of them are required, but Excel graphs are mandatory. Okay? Both of them are required. You have to submit both.
Sir? Hello?
Yeah.
Sir, last time you said that only an Excel graph was needed. So many of us haven't submitted the hand-drawn graph.
Only Excel. I don't think I said this. Okay, so
Yes, sir, it was like that. You said, yeah, we asked about like whether we have to draw draw graph with our hands, or just Excel graph is needed. So you said that no, only Excel graph is enough.
okay, so for last time, for this experiment, last experiment, please ignore this. It's okay. But for upcoming all the experiments, we have to submit both.
Okay, sir.